All right, so today we want to cover the following topics. We're going to talk about the phenomenon of interference between crossover events. And then I want to briefly introduce different types of polymorphisms in the human genome and use that to motivate introduction of another probability distribution, the Poisson distribution. And then I want to talk at the end about the application of the Poisson probabilities to mapping functions. All right, so here are my notes from the end of last class. We got a bit delayed last class, so I just wanted to go back and briefly go over this again. So let's see, can I take all right, so what we were talking about in the last class towards the end of the class was doing 3-point crosses, okay? So this is where you have 3 factors. So whereas, with Mendel, we were talking about 2 factors and dihybrid crosses, now we are talking about 3 factors and doing 3-factor crosses. And I use the example in maize of having 3 different mutant phenotypes, the lazy, glossy, and sugary phenotypes. These are the mutant phenotypes. So we use the lowercase, LZ, GL, and sugary. And we talked about making a F1 heterozygote, okay? That's heterozygous at all 3 loci. And so we have the following genotype of having one allele of one mutant LZ allele and one wild type LZ allele. So we write it like this. And the point was they all exist on the same chromosome, but we don't know the order. And we also don't know the phase. So we don't know whether along one chromosome we have the mutant LZ and the mutant SU, but we have wild type GL, or if we have all mutant alleles along one of the chromosomes and all mutant alleles along the other chromosome. And so what we're doing in a 3-factor cross is we're trying to identify what we call the phase of the alleles. Is it this case, or is it this case, or is it one of the alternative cases? And we're also trying to identify the order of the genes. Is it is it LZ, GL, then SU, or is it SU, then LZ, then GL? And we're trying to identify the distance between each of the genes, okay? So there are 3 different things we're asking to address in this. And so we do what we call a test cross, which is that we cross that F1 heterozygous at all 3 loci to a strain that is mutant homozygous at all 3 loci, because that allows us to only follow the gametes from one of the parents, which is what we're doing in this test cross, okay? Okay, and so what we get are data like this that come out of the F2, where we have, just based on the phenotype of the progeny, we can infer which genotype the gamete from the F1 parent that was heterozygous at all 3 loci we received. And so when we do this, we can immediately see that there are 2 prevalent classes. This genotype and this genotype are our most prevalent class, 286 of those, 272 of those. And that immediately tells us what the parental gametes were. And so now we know the phase of the alleles. We know that on one of the parental chromosomes, we had all wild type alleles. And on one of the parental chromosomes, we had all mutant alleles. The second thing we can see from this cross is the rarest class, where we only have 4 of these individuals and 2 of these individuals. And from this, we can infer where the double crossover occurs. And then we can immediately see the gene order, because we can see that this individual differs from the wild type by only the sugary locus. And this individual differs from this parental, again, at only the sugary locus. And so that tells us that sugary is the middle gene, okay? Because that is we get that swap, that difference from the parentals based on the sugary locus being recombined with respect to the parents. Okay, so this is what I reiterated here, is that in a genetic cross, the 2 most frequent types of the gametes are the non-recombinant or parental gametes. This tells us the phase, which is what we have determined here already, step 1. Step 2, the double crossover tells us which locus lies in the middle in this 3-factor cross. And so from this, we can see that it's the sugary locus is the middle genotype or middle locus. And then we can compute the recombination frequency between the LZ gene and the SU gene, and between the SU gene and the GL gene. And we do this by reorganizing this table and counting how many times we see recombination between LZ and SU, SU and GL, and remembering to include the double recombinant events, okay? And from that, we simply just compute frequencies to compute the recombination fraction. So in this case, recombination fraction between LZ and SU, that's 79 out of our 740, and between SU and GL is 109 out of our 740. And from that, we can make a genetic map. And this is our genetic map that puts the order of the genes along the chromosome, LZ, SU, GL, and then the distance between them, 10.7 centimorgans, which is simply a conversion from a recombination fraction of 10.7%. We use the 1% recombination equals 1 centimorgan. And so this is the other way this is how we write it. This is the 3-factor cross. And so now we have a genetic map that defines the relationship between these genes. And so let's just redraw that here, LZ, SU, GL. And we have the genetic distance between each of those loci, 10.7 centimorgans, which corresponds to a recombination frequency of 10.7%, or a probability of having a crossover of 0.107 per 1 meiosis, or 10.7 crossovers per 100 meiosis, okay? And so from this, we should be able to compute the expected frequency of a double recombination event. So if recombination probability here is 0.107, recombination probability between SU and GL is 0.147, then the probability, we just use their independent events. So we have independent probabilities, so we multiply them together. And we say that if they are independent, we expect the probability of a crossover at this location and at this location is 0.0157, multiplying them together, or 1.57% of our progeny should be double recombinant. And so that means that we expect to see 11.6 of the 740 individuals are going to be double recombinant. However, in our data, in our empirical data, we see 6. And why is this? This is because of a phenomenon known as interference. So what interference is the phenomenon in which a crossover here is going to reduce the probability of having a crossover in this region adjacent to it. And we can quantify this as simply 1 minus the observed number of double recombinants divided by the expected number of double recombinants. This is our expected number, and this is our observed number. And so interference is a really interesting phenomenon that we observe in our recombination fraction in the progeny. But what it is actually a molecular phenomenon such that if we have two loci, if we have a recombination at a particular site on the chromosome, it's going to inhibit a recombination occurring immediately adjacent to it. And the strength of that inhibition changes as a function of the distance. So the further away, the less interference there is, the closer the two loci are, the more interference there is. And this is turns out to be a really fascinating molecular mechanism that underlies this. Has anyone ever worked with C. elegans? But everyone knows about C. elegans, the nematode. And they are really interesting because they have a phenomenon where there is absolute crossover. So in that case, absolute interference. So in those organisms, there is only one crossover per chromosome. If you have a crossover on a chromosome, then there will be no other crossovers. And so that's a case of complete interference. And so different organisms have different extents of interference. But in the human genome, this phenomenon is observed so that this reduces the frequency of the observed double recombinants with respect to the expectation based on independence. So I want to use this, we've been talking about maize, but now I want to talk about, how we can apply these same concepts to genetic mapping in humans. So when we are performing genetic mapping in humans, we have two goals. We want to firstly identify where in the genome the genes associated with the hereditary disease are located. That's the first step, is to map it. We are literally trying to ask the question, if we know we have autosomal dominant inheritance, how many autosomes do we have in the human genome? Autosomes, 22 autosomes, right? Everyone remembers that, because Down syndrome is caused by trisomy 21 and it's one more than that. Our first question is, which of the 22 autosomes does the gene lie on? If we just guessed, we have a 1 in 22 chance of getting it correct. So our job is, as geneticists, is to be able to perform genetic mapping and to be able to assign a disease-causing mutation to a particular chromosome. That's the first step. And then we need to refine down that region and identify the actual gene and then the causative variant. So firstly, most genes that cause genetic diseases are rare, so they're only observed in a small number of families. So we have the problem of identifying them and all the problems we talked about at the beginning of the class, dealing with small numbers. Many of the mutant genes are recessive, so they're not detected in the heterozygous genotypes. We're dealing with very small numbers. We can't do controlled crosses. And then here's the key problem that I want to emphasize today, is that when Mendel did his experiments and looked at independent segregation of traits, and then when subsequent experiments were done that showed that there was not independent segregation of traits, in those cases, those researchers like Morgan with his mutants was generating mutants that had interesting phenotypes that could be followed in a cross. But unfortunately, in humans, there are very few traits that you can follow in a cross and therefore be able to say, my disease gene is co-segregating with this other gene that confers this other phenotype. Okay, and so we don't have that in humans. And so this was a major limitation to being able to perform genetic linkage mapping in humans that was solved in this, landmark paper published in 1980 in the American Journal of Human Genetics by David Botstein and colleagues. David Botstein was actually my postdoctoral supervisor and is a yeast geneticist by training and knew a lot about genetics from working with yeast and bacteria. And so when he looked at the problem of human genetics and addressed this problem of how do we perform genetic linkage analysis in humans, he said, well, we don't have phenotypes that we follow, but we do have polymorphisms in the human genome that we can follow in and look at how they segregate and ask, is there evidence for co-segregation of that genetic locus and our disease gene of interest? Okay. And this is a paper as aspiring human geneticists, you should all read. I'll maybe remind me to post this and you can take a look at this paper. It's really fascinating to go back and read this paper. But this really set the foundation for genetic linkage mapping in humans, which then allowed us to be able to build physical maps and ultimately sequence the human genome 21 years after this paper was published. And so in this paper, they used polymorphisms that were known at the time, which are called restriction fragment length polymorphisms, or RFLPs is the abbreviation. And what is an RFLP? Well, this is a type of polymorphism that can be detected using the state-of-the-art technology that was available at the time. And the state-of-the-art at the time were two things. One is the ability to digest genomic DNA using restriction enzymes. So these are the enzymes that you have encountered in your classes like EcoR1 and BAMH1. These are restriction enzymes that are cloned from bacteria that cut DNA at specific sequence sites. Okay, we can cut the DNA, and then at a particular DNA locus, we can say we know that there is a cut site at that locus, so we can probe for whether the DNA has been cut at that site. And then we can assay that using a method known as southern blotting. So who here has encountered southern blotting in molecular cell biology or principles of biology? This was a method developed by Sir Ed Southern that allows you to, through hybridization, to be able to detect a piece of DNA in the genome. So we have a probe that is specific to a particular locus in the genome, and we run on a gel the entire digested genome out on that gel, and then we probe for whether that cut has been made. And if there is a DNA polymorphism that changes the DNA sequence, we no longer get a digestion at that site because the restriction enzyme can no longer detect it. And so we can detect polymorphisms in the genome by virtue of this assay where we have either the presence of a restriction site or the absence of a restriction site, and we assay that using a restriction, using a southern blotting. And this was the method that was introduced in 1980 for being able to follow the segregation of genetic variation in a population. And what this allows us to do then is to be able to take an individual. So let's look back here at this individual. So in this case, we do not get digestion of at this site because there's a sequence variant that prevents the restriction enzyme from cutting the DNA at this site. And so our fragment is large, but at this site, we do get digestion. And so we get two pieces of DNA. And when we run them out on the gel, we can detect those two different pieces of DNA, which tells us the sum of these two pieces of DNA is going to be the same length as this piece of DNA because it's simply been cut. And that's indicated by the colors here. And then these are homozygous individuals. So this individual was homozygous for the restriction site. This individual was homozygous for the absence of the restriction site. And this individual is heterozygous. They have both one copy of the locus that doesn't have the restriction enzyme site and one copy that does have the restriction enzyme site. And so this allows us to be able to distinguish homozygotes and heterozygotes by using this approach. And so what this then allows us to be able to do is follow and ask the question, is there co-segregation between this marker, M, and this disease gene allele, D, okay? And so let's look at this cross here. So here we have what's essentially a test cross because we have the parentals where one parent is heterozygous for the disease, and they are also heterozygous at this polymorphic loci. And so you can see here, there's the heterozygosity. And then this parent is homozygous. So we only have one allele at the M locus, and they do not have the disease. They're homozygous for the wild type allele. And then the children are the F1s, right, from this parental crossing. And we can follow now and ask, is there co-segregation between the disease allele, the big D allele, and one of these, does it tend to be co-transmitted in the F1s with one of these alleles? And so let's ask what's happened in this first case. So in this first case, this child has, from this parent, everyone can see my cursor, right? Is that right? Okay, so we can follow it because the Zoom annotation thing is a bit annoying. Okay, so let's follow what's happening here. So in this child, they have inherited the little d allele from this parent, and they have inherited this large M1 allele at the M locus, okay? Does everyone see that? And now let's look at this next individual. So in this next individual, they have inherited the disease locus, big D, and co-transmitted with that disease locus is this form of the allele, the form that is cut and is digested. So everyone can see that here. And similarly with this individual, we have the same scenario. In this individual, when they inherit the little d allele, they again inherit the uncut version as they do here. And in this case, again, when they inherit the big D allele, it is co-transmitted with the cut version here. And in this case, we have transmission of the big D allele, but we have transmission of the uncut version. So this is an exception. So if we look down at the summarization down below here, we see, or this is our exceptional case, okay? So in all other cases, when you get the big D allele, like this individual, it is co-transmitted with this version of the M locus. and that happens here and here. And conversely, if this parent transmits the little d allele, that is co-transmitted with the uncut version. But in this case, we have transmission of the big D allele, but with the uncut version. And why would that, what is the explanation for that exceptional case?
Recombination? Maybe?
Recombination would explain that, right? If we look at that case, we can explain that individual by saying, well, maybe the big D disease allele occurs on a chromosome with this version that has the restriction site and is therefore cut, but a crossover would result in the disease allele being co-transmitted with the uncut version. And so we can identify that individual as a recombinant in our cross. This is a type of DNA polymorphism, and this illustrates the point that we can follow co-transmission of a disease state and a DNA polymorphism, and we can ask the question, is there evidence for linkage? Are they co-transmitted? And if there is evidence for linkage, we can also ask, well, is there evidence for recombination? And so we can ask two questions about, does it lie on that same chromosome? Does it tend to be co-transmitted with the polymorphic site? And if it does tend to be committed, at what frequency do we see recombination, which is going to be informative about the distance between the disease locus and that polymorphic site? And so in that case, if this is a useful approach for us to be able to solve that problem of looking at co-transmission of a disease gene and a DNA polymorphism rather than a mutant phenotype that we can assay, then what we need are DNA polymorphisms in the human genome to be able to ask the question about co-segregation. And so I'm just going to summarize, and I'll do this very quickly, the different types of DNA polymorphisms that are useful for doing this. So one class of DNA polymorphisms are called simple sequence repeats or microsatellite markers. These are dinucleotide or trinucleotide repeats that are polymorphic. They vary in between individuals. And so we can identify homozygous and heterozygous individuals and follow them in crosses. And that we can genotype using methods like PCR and gel electrophoresis to detect those differences in size that reflect the differences in the number of copies. And we can do the same thing. We can ask questions about co-segregation of a disease gene in this autosomal dominant case. And we can ask when individuals are afflicted with the disease, are born with the disease, do we see co-transmission of an allele at the M locus here? And you can see again that in this case, when individuals inherit the disease, they also tend to inherit the M2 allele here. And when individuals do not inherit the disease, they tend to inherit the M1 allele. So again, we can follow co-segregation. Has everyone heard of microsatellite repeats before? They were very useful for a number of years, but then they were subsequently supplanted by single nucleotide polymorphisms. So these are individual mutations that occur in the human genome. The human genome is 3 billion bases, and we're all 99.9% identical. But 0.1% of 3 billion is a very large number still. And many of these differences are single nucleotide variants, or SNPs, or single nucleotide polymorphisms. And these too can be used as genetic markers in linkage analysis, and they can be assayed using methods like DNA microarrays, or DNA sequencing now is another way that we can detect these polymorphisms in the human genome and follow their segregation and crosses. And we can, in fact, on this microarray, which was cutting edge in 2008, we were detecting 100,000 single nucleotide polymorphisms in genotyping individuals at a single locus. And now, of course, you can spit in a tube and send it to 23andMe, and they'll do the same thing for $99. And so this was all due to technical advances in genotyping technologies. So you've learned how these mutations occur in the human genome. They occur through processes like DNA replication errors or spontaneous lesions. And I have a summarization of those different mutagenic processes here, which just for your interest, but we don't want to get into the nitty-gritty of the molecular mechanisms that result in DNA mutations. We want to think about rates of mutations instead. And so SNPs occur, these mutations are introduced into the human genome at some frequency, but then luckily for us, or we'd all be riddled with mutations, we have proofreading functions that reduce the error, but there is still some mutations that come through, and so the rate of error is on the order of about 1 in a billion bases are mutated per DNA replication event. And so I want to think about the number of mutations that we can expect. So if on average, the probability that any single site in the human genome is mutated through the process of cell division, there's an extremely low probability that any individual site will be mutated. 1 times 10 to the negative 9, which is 1 in a billion. So we get 1 in a billion mutations per base per cell division. Very, very low probability that any base will be mutated. But we have 3 billion bases. We have many, many, many bases. And so if we have we have a non-mutation rate of 1 in a billion, but we have 3 billion bases, how many bases do we expect to be mutated in a single cell division? And we can simply multiply those together and coming up with the answer 3. All right, but here's the question. That's the expected number, right? Just based on the mutation rate and the number of mutations, we expect to have 3. But if we looked at 100 cells, would all 100 cells have 3 mutations? Yes or no? No? I like no. We expect to see 3 based on our known rate, but we know we're comfortable now as statistical geneticists. We know that if we expect to have 1/4 of children affected with carrier parents and an autosomal recessive disease, we expect, in a family of 8 that there would be 2 children affected, but we are very comfortable now with the idea that we could also get 0 affected children or 1 affected child, or we could have all 8 children affected, okay? And we can compute those probabilities using the binomial distribution. And so the question we have today is now, if we are looking at this scenario, if we looked at 100 cells, how many of them would have 3 mutations? How many of them would have 2 mutations? How many of them would have 0 mutations? And that is what we want to solve, the problem we want to solve today. And we solve that by considering this process to be a Poisson process. Who has heard the word Poisson before? Parlez-vous français?
It's French for fish.
French for fish, okay? This is a fishy process, but it's actually named for Monsieur Poisson, who was, so it's not named after fish. Unfortunately, could you imagine being called Mr. Fish? I'm not sure. In French, it sounds cool. So the Poisson process. So a Poisson process is, we talk about events that are occurring at some rate lambda. And so our rate is our mutation rate, 1 in a billion. And we talk about the events are independent in time and space. And so what the Poisson process does is it describes the discrete random variable, which is the count. It describes, the probability of having 1 or 2 or 3 mutations. And so let's look at the Poisson distribution. Who's ever encountered the Poisson distribution before? Yeah, it's extremely useful. The origin of the Poisson process, Monsieur Poisson, was a French man. He was in the military, and the French military at that time were in North Africa. These are the French legionnaires. And they had this terrible problem, which is that, some of their soldiers each year would get kicked in the head by a camel and tragically meet a tragic end. And so it was a major problem in the French military to ask the question, next year, how many soldiers do we expect to be kicked in the head by a camel and meet this tragic end? Because we need to plan for that occurrence. And so Poisson realized that this is a random process that's random in time and space. It occurs with some rate. And he figured out, I can tell you, the French military, the probability that we will have 0 legionnaires killed in this tragic accident next year. And this turned out to be a very important innovation in the history of statistical thinking. So the Poisson distribution, let me draw it for you. Well, again, let me just, set this up that what we have is we have n, our number of bases is 3 times 10 to the 9, 3 billion. And the probability that any single base is mutated is 1 times 10 to the negative 9, okay? But then if we multiply n by p, we get a value that we call our lambda, that's our rate, and that's 3. So when we multiply n and p together, even though we have a very small number and a very large number, when we multiply them together, we get a number that's reasonable, which is number 3, okay? And so the Poisson distribution describes our expectation, and the way we write this is that we say the probability based on the Poisson distribution for n events, we say is e to the negative lambda, that's this lambda here, multiplied by lambda to the power of n divided by n factorial. Okay, and this is our formula for the Poisson distribution from the Poisson process. And everyone's comfortable with factorial, okay? Everyone knows how to do exponents. Does everyone know what E is? The mathematical constant E? Has anyone got it memorized to 7 significant digits? 2.718. I think that's good enough for government work in this case. But it's a good little factoid to have. But we're certainly knowing that it's 2.718. I think everyone should know it to 4 significant digits, right? That's not too much to ask. All right. And so if we have that variable now, we can use this formula to address this question, which is to ask what is the probability of having 0 mutations. And so why don't you try and do this calculation yourself? I'll show you the first example. Here we go. Let's fill out a little table here. And let's say the number of mutations is here. And let's compute the probability of having 0 using this formula for the Poisson distribution. And so we would simply say, well, the probability of 0 using this formula is going to be e to the power of -3 multiplied by 3 to the power of 0 divided by 0 factorial. And what does that equal? And you're all freaking out now because you're saying, well, 0 factorial is 0 times 0, and that means there's a 0 in the denominator, and so we cannot compute. But you have to recall that 0 factorial by convention is defined as 1, is equal to 1. And so you can relax and everything's okay, okay? And we can do this calculation. 3 to the power of 0, what does that equal? 1, thank you. So we have 1 over 1, that's good. And so we just have e to the -3, and e to the -3 is 0.0498. So we have now computed the probability of having 0 mutations as 0.0498. That's about 5%. Okay, so now we can answer our question that about 5 out of 100 cells are going to have 0 mutations. Excellent. Why don't we do this for 1 mutation? Why don't you compute that yourself and using the same formula, and we can see if we get to the same answer. All right, simple. So who knows the probability of 1 mutation in the human genome when there is 1 cell division? 15% or 0.149? Very nice. What about the probability of 2 mutations? 0.224, we'll take it. What about the probability of 3 mutations? Oh, also 0.224, interesting. 4 mutations, 0.168. 100 mutations, well, you can do that, okay? So you can see that this is, we can calculate these probabilities. And this is the calculating the probabilities based on a Poisson process. And what's really interesting is we know that the average rate is 3, but in fact, the probability of having 3 mutations is 0.224, but also the probability of having 2 mutations is 0.224. Okay, that's interesting. And we could calculate the probability of having 100 mutations if we really wanted to. It's probably going to be something vanishingly small. But this is a property of the binomial, Poisson distribution, is that it's unbounded. We could compute the probability of having 1,000 mutations or 10,000 mutations. So it goes to infinity. And unlike the binomial distribution, which is a bounded distribution, but it is a discrete distribution because we're talking about 1 mutation or 2 mutations or 3 mutations. This means it's discrete. We're not talking about 1.5 mutations, for example, okay? And so because, I'm preparing you for the possibility that you are stranded on a desert island with no computer and no access to Google or R at some point in your future, I just want to make sure that you know that there's a shortcut, if ever you need this, in which you can compute the probability of any number as being equal to the probability of the preceding number of events, x minus 1, multiplied by your lambda over x. So your lambda is your rate and x is the number. And so this is very useful because this means that if we wanted to compute probabilities on our desert island, well, all we need to do is get our 0 probabilities. The probability of 0 is going to be e to the negative lambda. And once we have that, then we can do the probability of 1 event is going to be equal to the probability of 0 events multiplied by lambda over 1, in this case is our x, using this formula. And so we can compute now the probability of 2 events, the probability of 3 events by hand on our desert island. The other interesting property of the Poisson distribution is that the mean is equal to lambda, and the variance of the distribution is also equal to lambda. And so the mean equals the variance in a Poisson distribution. So as the mean increases, the variance increases as well. And here is our Poisson distribution, which you can plot in R using the functions that you're becoming familiar with. And now we're plotting the probability given a rate of 3 mutations per cell division, an average of 3 mutations per cell division, we can compute the probability of having 0 mutations or 1 mutation, 2, 3, 4, and so on. Okay, and this is our discrete probability distribution. We can also plot cumulative distribution. So you should be getting comfortable for switching between probability distributions, cumulative probability distributions that tell us what is the cumulative probability of having, for example, 4 or less mutations or 4 or more mutations that we can read off these cumulative distributions. And as I said, it's discrete like the binomial distribution. So we're talking about 1 or 2 or 3 mutations, but it's unbounded unlike the binomial distribution. So in principle, it can go to infinity. And so now what I want to use this Poisson distribution for is to go back to our crossovers. So we've been using it to compute the probability of a specific number of mutations in the human genome. But I want to use it to be able to think about the probability of a crossover between any 2 loci. So if we think about gene A and B in a genome, then what we look at is what we're able to identify as geneticists is the recombination fraction. So that's the proportion of the gametes that are recombinant. And what we're trying to actually look at is, or infer is, did a crossover occur in the gametes? And so the direct way to do that would be actually to go and type the gametes, to type the sperm or the eggs, and to be able to identify, is there actually a crossover at that event? But what we're looking at is typically the genotypes in the offspring and in making inferences about recombination in the gametes. And we use the following terminology that we talk about. 1 map unit is equal to 1% recombination because we're building genetic maps. So this is the unit that we use. And what we have defined 1 centimorgan with this symbol as 1% recombination. And these are different ways of being able to say the same thing. So we talk about 3.1 centimorgans, or 3.1 map units, 3.1% recombination, or a frequency or probability of recombination of 0.31. And what this means when we use this terminology is that there are 3.1 recombination events that are observed per 100 meiosis. But there's an important distinction that we want to make clear, which is that the map distance is the distance between two genes that equals the average number of crossovers that take place in the region per meiotic cell. So it's a measure of crossing over, of this molecular process of recombination crossing over. Whereas the recombination fraction between two genes indicates how much recombination is actually observed in a particular experiment. So it's a measure of recombination. And so there's a distinction between them. But what a map function does is it allows us to use the recombination fraction, the recombination frequency, to infer the number of crossovers. And that is what we want a map function to do. When we have complete interference between two markers, then our map distance is equal to the recombination fraction. Okay, so if by having a recombination event, a crossover event, we inhibit an additional recombination event adjacent to that, then we are going to observe those recombinants in our progeny, and that will be informative about the map distance. And this is certainly the true when the recombination fraction is low, when the genes are close together. However, we have this issue of double crossovers and multiple crossovers, which, as we saw in the last class, is going to allow us, or the result will be that we'll observe non-recombinant progeny. And so this tells us that the map distances that are based on the recombination fraction are actually underestimates of the actual crossing over frequency between two genes. And so let's consider how we can use the Poisson distribution or Poisson probabilities to estimate the number of crossovers from the recombination fraction. Okay, so let's start firstly by thinking about Thomas Hunt Morgan's approach. So Morgan said that when we observe a recombination, and I'll use the symbol delta for that, so we observe a recombination fraction, that this recombination fraction is informative about the map distance between two genes. And so we simply look at the number of recombinant progeny, and then we say, based on that recombination frequency, we convert that to a probability of recombination between those two genes. But what we want to actually do is, the goal is to use that recombination frequency to make a statement about the physical distance between those two genes. And so when the recombination fraction, for example, is, so if, for example, the recombination fraction is 0.06, that corresponds to a recombination frequency of 6% of our progeny will be recombinant. And that's actually a good estimate. So we can say, therefore, the map distance, the physical, or the map distance, the genetic map distance is 6 centimorgans, or 6 map units, are all synonyms. But as we said, as that distance becomes larger, then the probability of having a double recombination event that will undo that first crossover, and so we see non-recombinant progeny is going to increase. And so our ability to be able to estimate the actual map distance is going to decline. And so how do we solve this problem? So the way we solve this problem is, let's think about our two genes, A and B. And we'll introduce another character in the history of genetics, Haldane. And Haldane proposed the following. He said that the recombination between any two loci is a Poisson process. It's just the same as getting kicked in the head by a camel. Happens at some average rate, but it's independent in time and space, and that is what a Poisson process. So let's consider the following case. So if we have M crossovers between A and B, M is our number of crossovers, okay? Then using the Poisson formula, we can calculate the probability of having 0 crossovers, which if we go back to our formula is going to be e to the negative m multiplied by rate, which is m to the 0 divided by 0 factorial, and this luckily disappears because that's equal to 1. And so the probability of having 0 crossovers is e to the negative m. We can then say, okay, what about having not 0? So what is the probability in that case? The converse question to ask is, what is the probability of having 1 or more crossovers? So that's just asking the question, what is the probability of not having 0 crossovers? And of course, as statisticians, we now know that that's going to be 1 minus the probability of having 0 crossovers, which is simply 1 minus e to the negative m. And so Haldane proposed a modification of Morgan's mapping function, which maps the frequency of recombination is equal to the map distance. And he said instead, well, actually, the frequency of recombination, or the recombination fraction that we observe, is equal to the probability of there being 1 or more mutations, 1 or more crossovers, so the frequency of non-zero crossovers, multiplied by a half. And this is Haldane's mapping function. And so let's consider the case if we observe recombination fraction in our progeny of 27.5% of our progeny are recombinant, or as a fraction that's 0.275. Then we can simply use this formula. So we now say, okay, 0.275 is equal to half 1 minus e to the negative m. I do this calculation now, so e to the negative m is going to be 0.45, and therefore m is equal to 0.798. And now I can compute the probability of m crossovers, or on average. Therefore, on average, there are 0.798 crossovers between the two loci using this formula. So, but the question you're asking is, well, where does the half come from? So we have the number of crossovers. That's either, we have the probability of having 1 or more crossovers, but where does the half in that formula come from? And where that half comes from is that when we think about a single recombinant event, a single crossover, that results in 50% recombinant gametes. So we have the sister chromatids, and we have a crossover event, and we're going to get recombinants, okay? But that's only if we have a double crossover. Instead, we're going to see all non-recombinants if we're considering these two dashed lines as our two loci that we're following. But that's only the case when the two same sister chromatids are involved. But in fact, there are 4 players here, right? There are 4 copies of the chromosome. So actually crossovers can occur between any of these pairs. And so if we involve 3 chromatids in a double crossover event, we're actually going to have 50% recombinant gametes. Similarly, if we had all 4 of the sister chromatids are involved, then 100% of the gametes are going to be recombinant. And so it's actually the case that the 3 types of double crossovers occur in random proportion. So the average effect of a double crossover is to give 50% recombinant gametes, okay, if we consider all these possibilities. And so you can work this out, draw these pictures at home to consider what would happen in the case of a triple crossover, where we incorporate all possible triple crossover scenarios for having the 4 chromatids. But I will tell you that on average, what you're going to see is that any number of crossovers results on average in 50% recombinant gametes. Okay, if you draw this out, and that's where the 50% in Haldane's mapping function comes from. And so here's the point: we want to use the recombination fraction, which we'll define as theta, to estimate the number of crossovers m to determine the map distance. And because some crossovers are undetected, because of multiple crossovers, some of those products will not be observed as recombinants. And so what Haldane's mapping function aims to do is to account for these multiple crossover events. And this is the formula that we used. And so just to remind you that, we're looking at recombinant progeny and making inferences about these crossovers, which are actually these fascinating molecular events that we can detect in meiosis, these chiasmata that you can see under the microscope. Or you can stain using different stains. And so here we're staining with a protein that localizes to the crossover events. And you can actually look under the microscope in male gametes and count how many crossovers there are. And each crossover is going to produce 2 recombinant, 2 non-recombinant chromatids. So we're going to have 50% recombination. We know that 1% recombination equals 1 centimorgan. And so one crossover contributes 50 centimorgans to the genetic map length. And so that means we can actually look under the chromosomes, see the number of crossovers, the number of chiasmata, and multiply that by 50 centimorgans. And that's going to give us the total genetic map length for the genome. So if we look in humans, we see about 50 or so of these chiasmata on average. Multiply that by the corresponding recombination fraction of 50 centimorgans. And so this gives us a total map length in the genome of about 2,500 centimorgans. Interestingly, female humans undergo more crossover events. So actually there's greater recombination in the female meiosis. And so there's a larger genetic map length in the female. We've now learned, in fact, that if we look along chromosomes, that there's crossovers are not randomly distributed across along the chromosome in males and females, but again, there's greater recombination in female meiosis. And this also varies between individuals. If we look at different individuals and look at the number of recombinations per meiosis as measured using microscope method that allows us to look under the microscope and see the number of recombination events, that also varies between individuals. And so the last thing I want to do before we stop today is just to complete our calculation. So I just want to go back to this calculation now. We know that we have now, let's say, now we want to correct the map units. We want to convert back to genetic distance, correct the map units. We know that we have, let's say, 0.8 crossovers between these two loci. We know that one crossover contributes 50 centimorgans. So now what we want to do is ask, what does 0.8 crossovers equal? And that is a simple calculation. We just do 0.8 times 50 centimorgans equals 40 centimorgans, okay? And so whereas if we have a observed recombination fraction of 20.5%, whereas Morgan would say that that corresponds to 27.5 centimorgans, when we use Haldane's mapping function and convert back to genetic distance, we have corrected for the possibility of double and triple and quadruple crossover events, estimated the number of crossovers between those two loci and converted that back to a genetic map distance. And now we have a genetic map distance of 40 centimorgans between those two individuals. And those are our corrected genetic distance using Haldane's mapping function.